delighted to introduce our next guest, Amir Saffari, the Senior Vice President of AI BenevolentAI, who's going to be giving us a case study presentation now and walk us through how BenevolentAI is scaling and tackling the fundamental challenges in drug discovery by developing innovative AI technology. Amir.
Hello. Hey.
Hey, give him a round of applause, everyone. Thank you.
Cool. Can you hear me? Cool. Are you guys feeling okay? After heavy lunch? Cool. So I'm going to talk to you about what we do at Benevolent. And I had a long title called Scaling and Tackling Challenges in Drug Discovery, but as I developed the slides and I developed the content, I think one thing that resonated with me, which I thought that should be the corner of the center of the talk, is around what we call AI augmented scientists. And I'd like to walk you guys through this. So when it comes to advances in AI, many of us translate that to automation, to taking completely over the jobs, that cognitive jobs that humans do, to job losses and all the horrible things that is going to happen to us. But Benevolent, we have a very different view to this, which we call AI augmentation, where we think about where it makes sense to augment a scientist with advanced AI technology and where it makes sense to automate. I think many of you are familiar or have heard about Kasparov's epic battle of chess with Deep Blue, where he got defeated, and it was the first time that a grandmaster was defeated by an algorithm. But I think what is less known about Kasparov is that after he was defeated, he went to work with computer scientists and AI researchers to work on a topic called advanced chess, where a chess player is working together as a teammate with an AI algorithm who's playing chess as well. And the combination of the two are the team that actually plays the chess. And what happens is that when you put these two pieces together, they become undefeatable. They are one of the best players in the world of chess at the moment. And we have the same philosophy when it comes to looking to scientific challenges as well at Benevolent. I think when you think about humans, we are very good with tackling problems, having intuitions where there's very little data. We often have wild imaginations. We can go off and generalize beyond what is really available there, which is great. But at the same time, we also often draw conclusions out of where there's no data to support it, where we see patterns in randomness, where there is correlation, where none exists. So computers, on the other hand, can augment us with grounding us to the realities, to the facts, and makes us become more objective in our decision-making process, be more data-driven, and ultimately amplify our intelligence. Another way of looking at it is to a law called Amara's law, where it describes how the technology impact pans over time and how we as humans expect of technology, which has a weird correlation or characterization to it. Where early on, I don't know if this laser would work. Yeah. So where early on, where the technology is still in development, early phases, we as humans often overestimate what the technology is capable of doing. We can generalize, we go beyond what it's capable of, and we often think that it can do much more than it's capable of doing. While after a while, we enter this phase of development where the technologies actually can do a lot more, but our expectation of imagination runs out of fuel. And I think if you think about the full automation, where we are automating completely, let's say, the discovery process, versus where we can augment our scientists with good technology that can amplify their creativity, we can see this differentiation, in my opinion, and this is where we are at this point in time, where our tools are very good at bringing our scientists to a level that they can be very, very productive, can go through hypotheses very, very quickly, can generate them, can test them, while I think the automation is still quite behind in the curve. Another aspect of how we look into the discovery process at Benevolent is through a holistic view of the entirety of the journey, an end-to-end journey going from data to medicine, to drugs at the other end. And I think in many of your organizations, you are faced with this, similar challenges, but often at a very, very big departmental level where, people that are doing your target identification are not talking to people that are actually doing the chemistry and designing your compounds, and people that are running the development, the clinical processes. they have no clue on where this actually compound came from. So you have to have a very holistic view into this whole process and bring all of that together to be able to effectively reduce the time to get from data to medicine. And I will talk to you a little about individual steps that we do as top progresses, but you have to have this holistic view, in my opinion. The other the other aspect of it is as well that it's not a linear chain. It's not that you start at one end and you basically push a cart forward in a line, and at some end there comes out drug. It's a very, very iterative process where, as an AI augmented scientist, you run a lot of in silico hypothesis generation, compound generation. You do a lot of experimentation, which can be done computationally. You take it to labs, you test them. Once your evaluation data is coming back, you take that, put it back into our knowledge graph where you have all the data in the world, train AI models again, and repeat. And if you can do this fast enough, and if you can do this as described previously in the previous talk, in a parallelized way, then you have a very good chance of reducing the time that takes us to come from an initial idea to where we have a good candidate that we can take to the development process. Since my talk was named under case study, I'm going to also give you guys a bit of a background on one of the programs that we've been working on for a while now, which is on ALS. So I think we started working on ALS back end of 2016, where basically we picked the ALS disease not because we were experts in it, or not because we had expert scientists that have been working on ALS for a long time, but actually for the opposite effect. We had developed a technology up until that point that we're able to ingest all the data, contextualize it, run machine learning models on top of it to draw hypotheses. But we have never done anything with ALS, so we thought that this is a perfect disease actually to start with. So ALS is a very, very challenging disease. It's incurable. And the life expectancy of patients is very little. The standard care is very poor as well. So it adds roughly about 3 months to life of patients. So it's a very, very challenging and difficult disease to work in. In order for our hypothesis to be tested, we teamed up with an academic group called CITRAN, who are the world-leading group analyzing in ALS. And they have developed assays that are quite challenging, but also quite representative of the disease, so that when we have hypotheses, when we have ideas, we can go there and test it with them. So one of the technologies that we have developed at Benevolent called Biome is Nlp Driven Technology, where we have machine read all the literature in the world. We have ingested all the data sets that exist. And we have contextualized and brought them all together in an intuitive platform that the scientists can easily start starting their research in a disease area. And for many of traditional scientists, it takes them months, maybe years, to become experts in a disease area. For our scientists, it's a matter of a couple of weeks, because all the data that they need, all the papers that they need to read, it all has been read, it's all been ingested, all the analytics have been done. So in a click of a button, they have access to this huge repository of information that they can get familiarized quite quickly with the disease area. Another area that we've invested heavily into it is what we call patient stratification, and bring it early on also in the journey of discovery, where rather than thinking about clinical design at the end where you're trying to think about your patients, we actually start thinking that early on, identifying what are your patient subpopulations, thinking about how we can characterize them, how we can model them. In this case, we use deep generative models to do this. But identifying that actually who are my target patients that I'm going to actually treat with this program. And these gives us initial stratification of patients that we can then take to next stages. Early on in back in early 2017. So by use of our AI technology and our inference algorithms, we selected 5 molecules out of the ones that has been suggested through our systems. We took these molecules to Citran to be tested. One of the molecules that we had didn't do much at all. 3 of them did as well as standard care, so that was good. But one of them was significantly better than what was out there. And this was very exciting, both for Citran, because they've been working on this for decades. And this was very, very a big breakthrough for them, as well reassuring to us that our models are actually starting to make sense of all this data and are generating hypotheses that actually make sense in the real world. Of course, as I mentioned, our tools develop, our data sets develop, and we can rerun these experiments any day. So over time, we did that. We experimented more, we generated more hypotheses, and we took them to labs again for testing. One of the areas that we have invested heavily in terms of our technology is our relation inference systems, who are able to create novel hypotheses, as well as, again, having scientists in mind, having intuitive explanations of what they do. So often, many of the machine learning models are what we call black box models. So they give you predictions, but you actually don't know why they are making these predictions and why it is happening. So in our systems, we are able to, with various ways, either through methodology we use, or through, for example, in this case, use of literature and a clever way of realizing that, give our scientists clues about why this target, for example, has been picked for the disease that you are researching. So from the new, there's a delay. So this is not clicking through, sorry. OK, cool. So from the new discoveries that we've made, so we've identified a new compound, which again, in the assays developed by Citran, they show about 10x more potency. So compared to our early discovery that we had, you can use 10 times less concentration of this compound in the same assay to the same effect, which is again hugely surprising, as well as very, very encouraging to us. The next step that we took for our ALS is to go through compound design. So now that we had our targets, now that we had our initial compounds to start with, we wanted to develop our own compounds as well. Our chemistry platform has 2 big components to it. One of them is called AutoChem, which is a compound property prediction system. So it's using deep learning models to model how the compounds behave and what are their properties. So as a chemist, when you use our platform, you can easily draw a compound and get all the predict properties for what you care in a second. But when you put that together with another platform that we have, which is called EvoChem, which is using deep generative models together with reinforcement learning, you can start designing compounds that are novel, that satisfy the properties that you want, and you can do this every day, day in, day out. So in the context of ALS, we did exactly that. So we took our property prediction models, put them in a multi-object parameter optimization framework, ran our EvoChem sessions, and what we had are these 2 new compounds that at the moment are in testing, which again shows a lot more rescue in motor neurons survival compared to our original idea, as well as the latest discovery that we had. So I'll close with also this topic that it's not also just about technology and putting it the right framework for scientists. It's also the way that the company works that matters. So this is how our design meetings at Benevolent look like. An AI researcher sitting with a chemist, working with the chemoinformaticians, and probably many of them together at the same room. So it's really a cultural shift in terms of how you work and how you design your teams, how you design your company, not just the tools and technology. It's much more than that. So I'll close with this slide, and I have time for questions.
please join me in thanking Amir for that presentation. Thank you very much. Can I keep you for a couple of questions? Is that okay? If you'd like to ask Amir a question about all that you've just seen there, please do raise your hands. Any questions from the floor? Hi, gentleman just there at the front. Anybody at the back this side? Hi.
Hi, thank you for the talk. I'm just wondering, the 5 candidates you selected in the beginning, could you elaborate on conceptually why did you think these 5 are the ones you want to test?
So they came through our hypothesis generation algorithms, models, that they rank hypotheses based on what they think are related to the disease. And they were at the top. Our scientists looked at them. They were quite well from their perspective. When we put them into experimentation, actually just one of them didn't work. But the next 3 were as good as what was out there. So that was also a good outcome. But one of them worked much better. Sorry.
Have they been tested previously in ALS-related assays, or have they been tested in other type of biological assay systems? How did you come up with the compounds in the first place?
So the way that our inference algorithms work is that we have machine-read all the literature, so we have extracted all the information from literature. We have all the databases that are in the bioscience domain, all linked up together, again, ingested into the same system. Our inference algorithms run on top of that database, which is represented as a knowledge graph. And what they do is that they try to predict what are the missing links from this knowledge graph, what are the relationships that we haven't discovered so far, we haven't written about it, or we haven't documented them in our databases. And those are the hypotheses that we then take to testing.
Okay, thank you very much. Thank you, Amir, for that and for your time as well. And should anybody want to ask Amir any further questions, you're here with us for the rest of the day and tomorrow as well?
Yeah, absolutely.
Wonderful. Thank you so much. Amir Saffari, thank you so much.
Thank you.